Okay, so can everyone see my screen? Can you still see the presentation? Okay, good. Are we good to go? Is there... Yeah? Yeah? Do I assume we're good to go? I mean, not that you guys would know if we're good to go, but yeah. Everyone sit with water and a snack or something because this is going to be really long, and I apologize, but that's just how it is. I am trying to record it, yes. I hope it works this time, unlike Geo, which I feel really bad for because it didn't work last time, but it should work. Yes, you should. I support that. Okay. So, this is Unit 8, people's most hated one. I don't think this is the most hated one, but this is one of the worst ones, I would say, even personally. So, the first part is fairly easy. It's about enzymes and activation energy. Assuming some of you take chemistry, you're probably going to know what activation energy is, and all of these definitions are more simpler, but it doesn't matter because it's just bio, so you don't need to know everything. Activation energy is the energy required to break the bonds within the reactant that allow it to be converted to products. So, when a reaction is occurring, energy is given out as new bonds are made because it's an exothermic reaction. When you make new bonds, energy is always released. That's why most biological reactions are exothermic, because it's always something will be breaking down, but then new bonds will be created. Next is that the enzymes reduce the activation energy of the reaction. So if a reaction is progressing normally, it will have a certain rate. When you use enzymes, it basically reduces the activation energy, which means it reduces the energy required, which means that the rate will increase because it takes less time to reach that threshold of energy required. So therefore, enzymes are catalysts, and they reduce the activation energy of reactions, and therefore make it easier for these reactions to occur. So the way enzymes work is when the substrate attaches to the active site, it weakens the bonds within the substrate and then changes it to a transition state. So this happens in all occurs in all reactions. The substrate first gets weakened and then the reaction progresses. But when enzymes are involved, the transition state that it goes into has significantly less energy, and so activation energy is overall reduced. That means that basically, since enzymes are involved and they're catalysts, they'll just reduce the amount of energy needed to break down the substrate into its products, and therefore the reaction will occur faster. So that's what enzymes do. This is enzyme inhibition. There's two types of enzyme inhibition that you need to know. One's competitive and one's non-competitive. So competitive inhibition is when the inhibitor looks very similar to the substrate. So if you can look at the top right picture, sorry, I can't point because it's a whole Discord thing, but basically when you look at the top right inhibitor, you can see the shape of the inhibitor and the substrate, and they're very similar in terms that the portion that binds with the active site is pretty much identical. The top may differ because obviously the substrate and the inhibitor are two different molecules in themselves, but when it's a competitive inhibitor, it has some of the same structure, so it can bind to the active site. So when the inhibitor binds to the active site, the substrate can't bind. Therefore, it basically reduces the chance of the reaction progressing. If you remember, if anyone takes chemistry and remember the collision theory, you're basically just making it harder for the frequency of collisions that occur between the substrate and the enzyme are lessened because an inhibitor is introduced, and that will collide just as many times, basically. So what happens is that the inhibitor binds to the active site of the enzyme, and while it occupies the active site, it obviously prevents the substrate from binding, and therefore the activity of the enzyme is prevented until the inhibitor will dissociate, because eventually it will dissociate. Because it's a competitive inhibitor, the inhibition is not permanent, which means that after a while it'll basically go back to the inhibitor will leave the active site and the substrate can then bind and turn into products. And that's why when you look at the graph at the bottom, when you compare the rate of reactions as substrate concentration increases with no inhibitor and with a competitive inhibitor, it basically reaches the same point, maybe a little less. And that's because of the fact that this is not permanent inhibition, which means that it's going to reach the same point of at the same rate eventually, because there'll be enough collisions occurring that it's gonna occur at a relatively same rate. So the activity of an enzyme is reduced if a fixed low concentration of a competitive inhibitor is added, but as the substrate concentration rises, the effect of the inhibitor becomes less and less until eventually it is negligible. Oh, by the way, if anyone has any questions while I'm talking, and if they're like completely related to what I'm talking about, you can ping me in the chat. But if not, you can keep putting in questions there, and I'll look at it after a certain like topic is done or something like that. Okay, so the next is non-competitive inhibition. So obviously we talked about competitive, and now it's non-competitive. The way non-competitive inhibition works is completely different to competitive inhibition. Obviously, competitive inhibition is when the inhibitor looks like the substrate. In non-competitive inhibition, the inhibitor are not similar. The inhibitor and the substrate are not similar at all. So what happens is the inhibitor binds to the enzyme at a completely different site. In some enzymes, it's the allosteric site from the active site. So it's off to the side. So if you look at the top left picture, it's not at the active site, but the inhibitor works in a way where it changes the conformation of the enzyme, which, if you recall back to unit 7, I believe, and protein structure, it basically changes the protein structure and the conformation, which then causes the active site to be altered. So it can result in two things: either the substrate can't bind at all, or the substrate binds but can't be converted to the product because it doesn't bind correctly. Obviously, since this is non-competitive inhibition, its effects are slightly different. The activity of the enzyme is reduced at all substrate concentrations, and the percentage reduction is the same at all substrate concentrations. So if you look back here, in competitive inhibition, it results in somewhat the same rate of reaction at the end. However, in non-competitive inhibition, the rate of reaction between no inhibitor and with inhibitor is different by an equal percentage. And that's because, since it's non-competitive inhibition, it will collide with the same number of enzymes and basically alter the same number of enzymes or same percentage of enzymes throughout the reaction at all concentrations. Okay, before I go into metabolic pathways, are there questions? Let me scroll up. Everyone's just talking about 6 and 11 and stuff. Oh God, med school. I wish you all the luck in the world. 3 is genetics. Yes, Linux, that was my username. It's okay. What else? Does non-competitive inhibition permanently change the conformation? Yes. I believe so. Oh, by the way, feel free to correct me if I'm wrong at any point, because I could very well be wrong. Sometimes, oh yeah, okay. So sometimes it can be reversible, which I will talk about in end product inhibition. But in a lot of cases, it's permanent. When we talk about end product inhibition, then yeah. No, don't worry about the pings. Feel free to ping me if it's something completely related to what we're talking about. Do you have examples? So for competitive I don't have specific examples, but if you want to think about inhibitors, the best example I can give you is usually for non-competitive is heavy metals. So a lot of heavy metals like zinc or any transition metal actually will basically work as a non-competitive inhibitor usually. Oh, cyanide is also a good example. I don't know about a lot of competitive inhibitors. Sorry. Oh, fair. I don't think you need to. I don't think they'll ask you that in the exam. I believe it's not part of the syllabus to know exact like examples, but of course if you have them, feel free to use them. No, I still don't think you. Yeah, so isoleucine and threonine are consistent and they are part of end product inhibition, which I will talk about after, like metabolic pathways. So that is an example that you need to know, which I will talk about. But I don't think you need to know specific ones just for competitive versus non-competitive. But if they ask you, like in paper 3 for example, if they ask you how can you measure inhibition or something like that, then you can use heavy metals for most enzymes. Opiums are an example of enzyme inhibition. I don't know completely. IB does love it when you give an example. Yeah, sure, you can. Aren't most drugs just inhibitors? Yeah, a lot of times, yes. But the thing is, a lot of drugs are inhibitors, but they might not be inhibitors of enzymes. They could be inhibitors of receptors and other things that are not related to enzymes. So that's why you have to make sure that you know that it's specifically enzyme inhibition. I don't have examples right now, but I can look them up later if you want. But obviously, if you find one, just double check on the internet if it's an actual enzyme inhibitor and not just an inhibitor of any other function in the human body. Okay, I'll keep going because it's going to be long if I stop again. Metabolic pathways. So metabolic pathways are complete pathways to reach from the initial substrate to the end product. And metabolic pathways always have these features. So there's always an enzyme that catalyzes every single reaction. All the reactions have to occur inside the cells, and oftentimes they are usually in the cytoplasm or the mitochondria. Some pathways will either build up organic compounds, anabolic pathways, and some will break them down, which are catabolic pathways. So obviously you need to know anabolism, catabolism, and metabolism as part of unit 2. So anabolism is basically the breakdown of anabolism is the creation of complex organic compounds from their simpler states, while catabolism is the breakdown of complex organic compounds into their simpler states. Some metabolic pathways will consist of chains of reactions, while some have cycles of reactions. In this unit, we look at both. So the chains, a good example for the chains is glycolysis, which obviously we will talk about because it's part of cell respiration. And then Krebs cycle is an example of the cycles of reactions where you have the coenzyme A and everything involved with that. Both of these types of metabolic pathways joined together are what cell respiration is basically about. And also in the Calvin cycle, it's also similarly done. Okay, end product inhibition. In a lot of these slides, you'll see a lot of information, and I'm so sorry, but there was no way for me to break it all down into less information. I won't read out all of it. I'll try my best to explain it, and I will put these slides up later if you want to reference it at any point. Okay, so end product inhibition is when, so if you look back over here, at the end of the metabolic pathway, there's an end product. End product inhibition is when that end product is an inhibitor of the first enzyme. So when it goes from the initial substrate to the first intermediate, I don't know if you can see my cursor, but when it goes from the initial substrate to the first intermediate, there has to be an enzyme that's used. When that particular enzyme can be inhibited by the end product, that's when end product inhibition occurs. So if you want the exact definition, when the product of the last reaction in the metabolic pathway inhibits the enzyme that catalyzes the first reaction, it's known as end product inhibition. In end product inhibition, the first enzymes are usually allosteric enzymes. So enzymes that have two non-overlapping binding sites. So if you look in this picture here, they have one site at the top, which is where the substrate is binding, but then they have a diamond-shaped or a triangle-shaped at the bottom. So these are two different independent sites, and they're on the opposite sides of this enzyme. And the allosteric site is completely separate to the active site. So allosteric enzymes are enzymes that have two completely non-overlapping binding sites, one of which is obviously the active site, and the other is called the allosteric site. The allosteric site is where the inhibitor will bind to change the conformation of the enzyme. In end product inhibition specifically, the end product binds to the allosteric site and changes the structure of the enzyme. So if you look here again, from the first to the third step, you can see that the first, it has a normal substrate, so a normal shaped active site that binds to that specific substrate. But at the end, when the product of the first one, or the end product of the metabolic pathway becomes the inhibitor, it changes the shape of the active site, which makes it very difficult for substrates to bind. So if the inhibitor or the end product detaches from the allosteric site, the enzyme will return to its original conformation. So when someone asked, can non-competitive inhibition be reversible? In this situation, yes, end product inhibition is always reversible because the inhibitor can easily detach from the allosteric site. So the advantage of end product inhibition is that when there is an excess of the end product, it will eventually lead to an inhibition of the first enzyme, which will basically stop the entire metabolic pathway until all the end product is used up, and then it detaches, and then the metabolic pathway can start again. So this basically allows your body to control how much of the end product there is. This is really helpful in isoleucine and threonine dehydratase. Threonine dehydratase is the enzyme that is the first step in the production of isoleucine, and isoleucine is obviously the end product. Isoleucine, when it increases in concentration, will eventually inhibit threonine, and then after isoleucine is all used up, it will basically return the ability of binding to threonine dehydratase, and then therefore it will result in a continuous controlled pathway. End product inhibition is an example of negative feedback. So in the exam, you can be asked about negative and positive feedbacks. End product inhibition would be an example of negative feedbacks. Okay, so I already showed you this image, but if we want to go through it, basically, obviously the substrate can first bind, and then it goes through its own metabolic pathway, and then as soon as it reaches the end product, and there's enough of the end product that enough collisions can occur, eventually the enzyme will get binded with the end product from the allosteric site, change the active site, and then therefore stop stop the metabolic pathway at its source. So one important thing to note about end product inhibition is that it doesn't stop the metabolic pathway in any of the intermediates. It stops the pathway right at the initial substrate, so you'll just have more of the initial substrate. You won't have any of the intermediates created at that point. Okay, so anti-malarial drugs. This is just an example of how they're trying to make use of end product inhibition. So the malarial parasite, or Plasmodium, has evolved resistance to most anti-malarial drugs, which is why we consider malaria to be such a difficult thing to treat, because most of the anti-malarial drugs are most of the malarial parasites are now resistant to the drug. So you obviously need new drugs to be able to adapt to their adaptations. So the search is being made easier by bioinformatics databases. And in a recent study, 5,655 chemicals that may act as an enzyme inhibitor in Plasmodium were identified. So obviously there's 5,655 chemicals that we don't know whether they would be an inhibitor, but based on their molecular weight and based on their characteristics, they were identified as chemicals that could possibly be enzyme inhibiting for Plasmodium. So obviously these were tested with 9 different Plasmodium enzymes, also identified from another database of metabolic pathways. And then when they were tested, inhibitors were found for 6 of the 9 enzymes, and then these are now researched as potential anti-malarial drugs. So the process of end product inhibition can be really useful in fighting parasites such as malaria, because then it basically, if you cause inhibition within the parasite, it will stop function of something and then will therefore cause issues within the parasite, which will stop it from affecting you. Okay, before I go into cell respiration, do you have any questions about metabolic pathways? Because everything after here will just go completely haywire. We are going to discuss photosynthesis later, so I will come back to this. But Adelina, if you just wanna edit your message and ping me, so I can, when I reach photosynthesis, I can answer your questions at that point, and it's easier for me to find. So either you can just make sure that I can, you can post it again, or you can ping me. Do we have any questions about metabolic pathways, or should I keep going? I'm going to assume that means no. Okay, so cell respiration, the great world. Okay, this is everything that is before glycolysis, or actually just the definition of cell respiration is part of chapter 2, but we obviously do a lot more than that in HL, so I mean, I'm going to presume most of you are HL. Cell respiration is the controlled release of energy from organic compounds, carbohydrates, to produce ATP. Oftentimes, these organic compounds are carbohydrates, such as glucose, but they can also be fat, or there can be other substrates. But in our case, we only learn about cell respiration using oxidation of glucose. So cell respiration is basically the production of ATP using energy, which is released by the oxidation of glucose. When glucose is a substrate, the first stage is called glycolysis, the second is the link reaction, and the third, of course, is the Krebs cycle. The first thing we'll go through is glycolysis. So, glycolysis is the conversion of glucose into pyruvate, and that leads to a release of small amount of ATP. When it's anaerobic respiration, the pyruvate that's created from this step converts into lactate or lactic acid, and that in humans at least, and that's where it stops. But of course, we're going to be studying aerobic cell respiration, so we will learn about where it goes after glycolysis. Glycolysis, of course, has a whole pathway which is catalyzed by enzymes because it's a metabolic reaction. Glucose is partially oxidized and a small amount of ATP is produced. This partial oxidation does not require oxygen, which is why this step is in both aerobic and anaerobic respiration. Okay, before we go into glycolysis, there's some things that you just need to know. First is oxidation and reduction reactions in cell respiration. So in respiration, there's a lot of oxidation that happens with the substrates, and the reduction is happening to the oxidizing agents. So that's what it is basically. Oxidation of substrates oxidation of substrates in the context of respiration is the removal of pairs of hydrogen atoms, and the reduction of hydrogen carriers, or NAD in this case, is accepting hydrogen from the removal from the substrate. So NAD, or nicotinamide adenine dinucleotide, which by the way I don't think you need to know at all, so don't worry, you just have to say NAD, is the most commonly used hydrogen carrier. Now there's two ways to write down NAD. You can use either, but there's NAD and NAD plus, and then if you write NAD, your product will be reduced NAD, but if you write NAD plus, then your product will be NADH with another H proton left behind. Phosphorylation. So the addition of a phosphate group, which is PO4 with 3 electrons, to an organic molecule. So the addition of this phosphate group is known as phosphorylation. Phosphorylation also makes the organic compound or organic molecule less stable, which means that they're more likely to react in whatever the next stage might be. So an example of this would be glucose plus ATP. ATP is what gives the phosphate group. Will create glucose 6-phosphate and ADP. That's an example. So basically, phosphorylation will convert what would otherwise be a slow endothermic reaction into a rapid exothermic reaction. Okay, glycolysis. Glycolysis basically has 4 steps, actually 3 steps. Step 3 and 4 happen in the same process. If you look at the right flow chart or the right metabolic pathway, then oxidation and ADP formation happen at relatively the same time, but I've written them down as 2 different steps. So the first, of course, is phosphorylation. The example I used in the previous slide is what happens here. So the first step is glucose, or hexose, gets converted into hexose biphosphate with by adding 2 phosphate molecules from ATP to convert it into 2-ADP. Now this process is of course called phosphorylation, so it goes from hexose to hexose biphosphate. I'm going to say hexose for the purposes of this because it's a lot easier to write hexose and then hexose biphosphate if you think about it logically, since there's 2 phosphates being given over to a hexose molecule, which is why it's called hexose biphosphate. Um, in this situation, of course, the two molecules of ATP give the phosphate groups, and the energy level of hexose is raised due to the phosphorylation, which is what leads to the less stability, and then the subsequent reactions are possible. In lysis or lysis, however you want to say it, I would say lysis. Lysis basically means splitting. This is where hexose biphosphate, which is a 6-carbon molecule, converts into 2-triose phosphate, which means 3-carbon molecules. Now, this splitting, of course, is called lysis. It converts from hexose biphosphate to 2-triose phosphate. After there are 2-triose phosphate molecules, it goes through both oxidation and ATP formation to lead to 2-pyruvate molecules. So, 2-triose phosphate molecules each let go of one H molecule to get 2 NAD to be converted to 2 reduced NAD. In ADP, the 2-triose phosphate molecules get rid of phosphate and energy to get rid of energy to convert ADP to ATP. But in this case, it converts 4-ADP to 4-ATP. So if you look back to phosphorylation, there you're using up 2-ADP, but at the end you're making 4, which means the net ATP production is 2-ATP. The end product, of course, is pyruvate, which is then used in the link reaction. Does everyone understand glycolysis? To test you, can everyone, before you write it down, spoiler your message using the 2 double bars on either side of your answer? But can someone everyone, if you can, write down the summary of reaction? So I want to know about ATP, I want to know about NAD, and what's the other one? You guys will know. Is what I'm gonna say. Why creative? Why? Let everyone do it. There's like 20 people here, and usually there'll be like 3 people answering anyway. No, you have to answer this too. You've been here. Now you should know. Spoiler your answers before you answer, please. Just so you don't, so that everyone gets a fair chance. What do you mean you just got here? You should know what's happening. Come on. I mean, to be fair, you're a physics student. It's okay. Okay, Vinix, that's not a bad thing, but I want to know, like, specifically. So if I go back just for a second, I want to know what ATP converts into. I want to know what NAD converts into, and then I want to know what hexose converts into. Just a summary. I just want what the conversions are. I don't need to know every single small thing, but I want to know what each of the major players are. Creative, give it a shot. Come on. Do I have to send you the answer? No, I'm not going to send you the answer. You've been here. You've been here the whole time. It's okay. It's okay. I won't judge at all if you get it wrong. Don't worry. And I'll put the answer up in... Oh, you got called away. It's okay. You'll see the answer in like 2 minutes. Once the several people stop typing. So, don't worry. It's fine. Oh my god. Uh, double bars. So just you added one bar on each side, just add another one on each side. No, it's okay, don't stress. Just edit your message. Okay. Several people are typing. This is the first time I've had this much engagement on a session, which is fair because this unit is severely annoying. Streamer moment. Truly a streamer moment. By the way, how do you pronounce lysis or lysis? Lysis. Lysis? Lysis? I always say lysis. I've always said lysis. Because the problem is, I'll say electrolysis, but then when I talk about glycolysis and just talk about the lysis part, I'll say lysis. It's so bad. But again, honestly, you don't need to know how to pronounce anything as long as you can spell it. That's what I firmly say every single time. I've said it in every single revision session. It doesn't matter how you pronounce things as long as you can spell it. At least in the context of a test. Okay. Very good, Leaf. Also very good, Steph. Spoken like a true spelling bee master. I know, right? I actually won spelling bee when I was a kid. Surprisingly. Don't say I knew it. What is that? Just the energy I put out into the world? Okay, you got icicle, I'm gonna assume. You got everything right except the first part. It doesn't convert ADP to ATP. The first part converts ATP to ADP. That's the only one. Okay, phosphate and glucose, hexose biphosphate, lysis, hexose biphosphate, 2-triphosphate, oxidation. Yes, Ali, good job. I'll give you 100% on that. Okay. I'm gonna wait for Ari to be done typing before I reveal the answer. Has Ari answered already? Ari has not answered. Yib Yob. Do I just call you Yib Yob? That's a cool name. Yib or any variant of your name. What if I just call you Yob? Okay. Yeah, I was gonna ask, do you want me to go over glycolysis again if you missed it? I mean, I will talk about it, but yeah. Oh wow, okay. Phosphorylation. No, it's okay. It's okay. After being done with 11 exams, I would expect you to start losing your memory at this point. That's a very good answer, Ari. I would say that's a really good answer. The only thing... No, you got everything. I was gonna say the only thing you missed, but no, you don't miss anything. You're good. That's good. Okay, so of course this is a very basic one, but basically one glucose converts into two pyruvates. 2 ATP gets used per glucose, but 4 are produced, which results in a net yield of 2 ATP. 2 NADs are converted to 2 reduced NADs. Of course, this is a very simplified version. Everything that everyone else sent is a really good summary version. Just if you, because I feel like I haven't checked every single one, if you feel like you got something wrong, just let me know if you're confused about why you got it wrong. But of all the ones I've seen after, like, Missin, I think, has all, or Mitchin, have all been correct, I think. Anyway, let me know if you have any questions anyway. Just ping me. Okay, so, this is a break because I need water. The challenge is to draw alpha D glucose and send your drawings in the chat so I can check them. This is just for fun. You don't have to do it. But everyone go drink water and eat food if you haven't already. I don't know what time zones people are in, and I'm not going to ask. But you cannot look up a ref. That defeats the point. If you looked at a reference, no, that's why it's a challenge. It's fine. Just try your best. Send it in chat, and then you can look at a reference. Or you can just DM it to me if you don't want to send it in chat and avoid whatever you consider this to be. If you consider this to be public embarrassment, you can just DM it to me and I won't judge. Fair. That's fair. Your momos are delayed. I feel so sorry for you. Genuinely. Now it's making me want to eat momos. You've ruined this for me. I'm dying now. I have water. Actually, yeah, fine. Okay, I'll be back in 5 minutes. Please try your best to draw alpha D glucose so I can see at least, I want to see at least 4 drawings in chat. And one of them has to be from you, Kroot. Is he still here? That's what I was waiting for. What do you mean, why? One of them has to be from you. I just want to see what it would be like if someone who doesn't take bio at all tries to draw a glucose molecule. Here, for reference, C6H12O6, right? That's what a glucose molecule is. Try your best. It's okay, Ori, try your best. Genuinely, try your best. C6H12O6 is what you need to know, and then try your best to draw it, and then I want to look at it. I will be back. No, spoiler it so that they don't see. Ari, spoiler it. Delete it and then spoiler it. Or spoiler it. I don't think you can spoiler it after it's already done. Just delete it and then resend it, and then you can spoiler it that way. I love the freaking... It's fine. Just leave it up there at this point. It's okay. Okay, I'm gonna go get water. BRB. Okay, I'm back with water. Let's see. Okay. Leaf, I swear you sent it. I didn't I saw that you sent it before I left, but now it's disappeared. Put it back in chat. Okay. Down, down, up, down. H H O H H H. Who was this? Ari, good job. CH2OH. H. Down there. Okay. Oh my god, Kroot. Oh my god. Oh my god. I'm just gonna save this. This is gold. This is pure gold. Okay. Okay. Yob, not horrible. You got the basic structure, right? And you got where the O should be. You got CH2O. The only problem is O can only be bonded to 2, so there's going to be no H at the O. The carbon on the very right and the carbon on the left of that is correct. On the carbon on the completely left down side, there's going to be an OH on top. And on the complete left side, there's going to be an OH on the bottom. So, not bad. Not horrible at all. That's a really good attempt. For if you don't remember, that's a really good attempt. What was I going to say? Oh yeah, lavender. The only thing I'll say is I think you tried to make it 2 at the top, but I just can't see it. It is CH2OH. So, yeah. Kroot, I would appreciate it, but I have no idea what that means. So, yes, I am very happy. This is making me really happy. You've just given me a dopamine slash lavender. Lavender? I was going to say lavender. Dopamine slash serotonin boost. I'm happy. This is making me happy. It's because I was looking at Lavender's message right as I was saying it, so I just didn't see it. I think I don't know if that counts as rule 4. I'm not. That's fine. Kroot is the admin. He can decide. Yeah. Okay. Cool. I'm just gonna pretend I didn't see it. I mean, you can remember it in whatever way, as long as you're remembering it. So, does anyone know all the chemical configurations you need to know for bio? What do you chemical configurations as in structures? Yes, if you go into M24 biology and pings, there's a thing that has every single diagram that you need to know. So, and within those diagrams there are the chemical structures. Yes, chemical structures it's called. If you go into the pins, not pings, with I'll link it after the thing is done. I'll link it. Ali, that's completely right. You're good. It's all good. Okay. Are we good to go? Is everyone back from water break? I don't know if anyone else got water, but I did. So, there's that. Good job. Okay. When are your momos gonna get here? Now this is a story I need to know. This is so sad. I feel so sad for you. I'm overwhelmed with grief at your lack of momos. What happened to Abby's wife? Nothing. I just switched because this we created Animal Farm. It's okay. It's hard to explain. There's a lot going on. I am English HL. If that was a question for me, then yes. Of course, that's why I was part of the Animal Farm. Stricken with grief. Oh, lol. Yes, I am English HL. Crude, stop judging me. Thank you, Vinix. I really appreciate the support. I really do. Crude, stop judging me. It's funny. It's funny. You had to be there. This is I feel like I'm streaming now. This is my true streamer moment. You can't give subs. I feel like you're not ruining my revision session. I'm on break. It's okay. It's okay. We have 2 minutes more. No, I'm not gonna refer to you as chat. Please, God, no. Gift Nitro. Yeah, truly. That's how you sub. Sub with Prime means you give me a Nitro. Why is this turning into a stream? What's happening? Should I just start streaming about a bio? I'm not gonna. No. Crew? No. Yeah. I feel like I should start doing bio streams. Is everyone just gonna be here? Oh dear God. Oh dear. Okay, yeah. Okay. We're gonna keep going. Bye. I'm not looking at chat anymore. Okay. Difference between anaerobic and aerobic respiration. So part of this is in unit 2, the SL part. And oh dear God, I didn't call you chat. I need to stop looking at this, for fuck's sake. Okay. You know what's so funny? People who are gonna look at this recording later on are gonna be so confused because it's not recording my Discord screen, so they have no idea what's going on. They just think I'm rambling. It's gonna be really funny. Anyway. Okay. Anaerobic or aerobic respiration. Glycolysis can occur without oxygen. I talked about this before. So it forms the basis of anaerobic cell respiration. So if you look at the diagram, glucose through glycolysis will turn into pyruvate. When there's no oxygen, pyruvate will take a pathway towards ethanol and CO2 in yeast and plants. But in humans, it'll turn into lactate or lactic acid. Of course, if there's oxygen available, eventually it'll go through the entire process of cell respiration, which we will talk about, and convert into CO2 and H2O. Krebs or Calvin. No, stay here, Crude. Stay. It's so much more funnier. Please, I beg. Krebs or Calvin. I would pick, personally, Krebs. ETC is after Krebs. Okay, cool. Back to topic. Anaerobic and aerobic respiration. Like I said already, if there's oxygen, it'll reach completion of cell respiration, but if there isn't oxygen, it'll just go through glycolysis and then convert into ethanol or lactic acid. We don't have to talk about how that happens, we just need to know that it does happen. Pyruvate that's produced in glycolysis can only be oxidized further, obviously, in the presence of oxygen, because oxidation only more energy can only be released if oxygen is available. The next step occurs in the mitochondria or in the mitochondrion, if you're talking about singular, which by the way makes no sense to me, but we will talk about that in my next break. The first stage is called the link reaction, and then the link reaction later result in the Krebs cycle. Link reaction. So, the pyruvate from the glucose is absorbed by the mitochondrion, which, by the way, the glycolysis occurs in the cytoplasm, and then it gets absorbed into the mitochondrion. Enzymes in the matrix of the mitochondrion will remove the hydrogen and carbon dioxide from the pyruvate to convert it into an acetyl group. Of course, if you do chemistry, you might know what the acetyl group is. You don't need to know that for this. And you're already done with your chem exam. So I can presume you don't need to know that until the next time you do chem, which for a lot of us is never. So, basically, the pyruvate will turn into an acetyl group, which then attaches to a coenzyme A. But the process involves the removal of hydrogen and carbon dioxide. Obviously, the hydrogen is accepted by the NAD, which we know is the hydrogen acceptor or the oxidizing agent throughout this entire process. NAD will turn into reduced NAD. It'll take a hydrogen away. And the entire conversion from pyruvate to acetyl group is called oxidative decarboxylation, since you remove hydrogen, which is called oxidation, and you remove carbon dioxide, which is called decarboxylation. So the entire process is called oxidative decarboxylation. The product of oxidative decarboxylation of pyruvate is an acetyl group, which is attached to coenzyme A or CoA to form acetyl coenzyme A or acetyl CoA, which is then used in the Krebs cycle. So if you look at the diagram at the bottom, pyruvate turns into acetyl CoA because the coenzyme A gets attached, but carbon dioxide leaves, and NAD accepts a proton in the form of H+, which then eventually leads to an acetyl CoA group. That's the entire link reaction. The next one are everyone's favorite, the Krebs cycle. So, obviously, we talked about how acetyl-CoA is created. Those acetyl groups from the link reaction are fed into the Krebs cycle. So, an acetyl group is transferred from acetyl-CoA to a 4-carbon compound, which is called oxaloacetate. I can do this. Oxaloacetate to form a 6-carbon compound, or citrate. I believe you don't need to know the names, but if you know the names, you can put them in. Don't feel pressured to remember the names. As long as you know that it converts in an acetyl group gets transferred from acetyl coenzyme A to a 4-carbon compound and then converts into a 6-carbon compound. As long as you know that occurs, you're okay. You don't need to know oxaloacetate and citrate specifically. So within the Krebs cycle, there are 3 types of reactions. There's decarboxylation, oxidation, and ATP production. Decarboxylations occurs in 2 of the reactions. Carbon dioxide is removed, that's decarboxylation, and it is a waste product, which is excreted together with the CO2 that occurs here in the link reaction, and the CO2 that's removed from the reaction from the carbon compounds in the Krebs cycle, and the ones from the link reaction are all excreted together, and by excreted I mean exhaled out, but of course for the purposes of this we just called it excreted. The oxidations, of course, the oxidation in all of cell respiration is the removal of hydrogen. This occurs in 4 of the reactions, and instead of just NAD in this one, there's 2 different hydrogen acceptors or oxidizing agents or hydrogen carriers. So there's NAD or FAD. NAD occurs in 3 out of 4 of the oxidation reactions, and FAD is converted in 1 out of 4. Both of these go from NAD to reduced NAD or FAD to reduced FAD. You can also write NADH to NAD plus to NADH, or FAD plus to FADH, depending on how you want to write it. But for a lot of people, it's just easier to remember NAD, FAD, and then reduced of each. Both of these, of course, are hydrogen carriers, which become reduced. They accept the hydrogen protons, and as they accept the hydrogen protons, they release energy, which is stored by the carriers and then later released in the ETC, or the electron transport chain, which is used to make ATP eventually. So, of course, these carry the hydrogen carrier. They basically carry hydrogen until the electron transport chain, so that they can convert the thing into reduced NAD and reduced FAD back into NAD and FAD and release electrons. But we will look at that after the Krebs cycle. There's also ATP production. That's the last one. This occurs only in one of the reactions. This reaction is called a substrate level phosphorylation. Okay, so this is the diagrammatic representation of how the link reaction and the Krebs cycle occurs. Of course, from pyruvate to acetyl coenzyme A is the link reaction, and after acetyl coenzyme A, it's the Krebs cycle. Now, you will notice that this is a continuous cyclic process, and the reason it is that is because, of course, there's a coenzyme, which means that it doesn't get used at all. It's just used as a transport mechanism, and also the same the same six carbon compound is converted to the 4 carbon, and then again converted to the 6 carbon, and then again to the 4 carbon. It's a continuous cyclic process which continuously releases hydrogen protons to create reduced NAD and FAD. So we know that pyruvate converts into acetyl coenzyme A. Then that acetyl coenzyme A deposits the acetyl group and creates a 6 carbon compound. After that 6 carbon compound, there's 1 carbon dioxide that leaves and an NAD that gets reduced. That 6 carbon compound turns into a 5 carbon compound, where again NAD is reduced and CO2 is removed. As you can notice, in every single one, CO2 is removed, and then it reduces by 1 carbon in each step, which is why, of course, that's how deoxycarboxylation works. It goes from 6 to 5 to 4, and it stays at 4 because only 2 CO2 is released. So a 6 carbon compound releases a hydrogen and CO2, converts into a 5 carbon compound, releases another hydrogen and another CO2, converts into a 4 carbon compound, at which point ADP turns into ATP, and that's the step where, of course, some of the energy is produced, and then the energy is used to convert ADP to ATP. There's also release of 2 other hydrogen protons, one that is accepted by FAD and one that is accepted by NAD. Do we have any questions about the Krebs cycle? Officer, can I keep selling sex for money? Acetate, citrate, isocitrate, alpha keto glutarate. I'm not going to read the rest of that. Okay, I'm drinking water. Do I have to talk? Oh god, this is turning into a stream now. Okay, yes. What's the acetyl bind to form 6C? I don't know what that question means. What does the acetyl bind to? It just binds to itself. The acetyl groups are groups of 2 carbons each, I believe. and then 3 of those will bind together to make the acetyl group. Yeah, I talked about it here, right? An acetyl group is transferred from acetyl-CoA to a 4-carbon compound that already exists to create the 6-carbon compound. So basically, there's only 2 gets deposited into an already pre-existing 4 that obviously you can see here is being created at the supposed end of the sequence. If you notice, basically on the left of acetyl-CoA, there's 4 carbons, and then it gets automatically converted to 6. The 6 happens because acetyl coenzyme A deposits another acetyl group, and then it gets converted to 6 again, and then goes through the whole process, gets converted to 4, another pair attaches. So that's why it's a cyclic process, because that's how it works. I mean, you don't need to know the name, but yeah, the 6C is I don't think you need to know about that. You just need to know that there is a 4-carbon compound that's created at the supposed end of the Krebs cycle, and then that same 4 is then used again when acetyl coenzyme A deposits 2 more. I don't think you need to know the specific names. I can check it. Let me check it with my we are going to discuss photosystem at 1 and 2. That's going to come after cell respiration. I'm still on Krebs cycle. So we're going to go through Krebs cycle, and then we're going to go through the electron transport chain, and then we go through photosystem 1 and 2, and then we go through Calvin cycle, and then we're done. I am going to give you another break after we're done with the entirety of cell respiration, and then we will, and there's another challenge there, and then we will go through the end of all of photosynthesis. Okay. Any questions related to the Krebs cycle? Is there anything I didn't explain well enough? Anything that I can re-explain? There's no worries at all. Also, by the way, my accent will switch back and forth a lot, and I apologize for that. But yeah. That's just what happens in do I assume everyone's okay with the Krebs cycle? Are we good to go on? Jesus Christ. Why is reduced NAD repeated twice? Reduced NAD is actually repeated 3 times. It happens 3 times. NAD happens 3 times, FAD happens 1 time. That's just how many times it happens. That's how many how many protons are released. It happens that many times so that it obviously converts 6 carbons to 4 carbons, but then it also happens so that there's enough energy to use in the electron transport chain. But you basically, at this point, it's just remembering the fact that NAD happens 3 times and FAD happens once. In IB level biology, you don't need to know why it happens that many times. Okay. I'm going to wait another minute if there's any questions, because I'm going to drink water. But, yeah. Is the Krebs cycle in a form of a loop? Yes. So if you remember when I talked about, I'm going to go all the way back. When I talked about metabolic pathways, there is cyclic metabolic pathways, and there's chains of reactions. In the Krebs cycle, it's cyclic, or it's cycles of reactions, but in the link reaction or in glycolysis, they're both chain reactions. They glycolysis and link reactions are both have a chain of reactions, and therefore they're the chain metabolic pathway. But Krebs cycle, obviously, since it keeps going in circles, it's the cyclic form of metabolic pathway. Anti-malarial drugs. It's basically an example of where what's it called? End product inhibition can be used. Like we can force it to be used. It's just an example. I don't know if they've ever asked questions about it, but I saw it in the study guide, I believe, so I just put it there anyway. That is the electron transport chain, and that's the electron transport chain for photosynthesis. We will get to that eventually. Missin. I feel like I've done this entire thing before. Okay. Are we good? Good to go? I'm gonna presume we are. If there's any questions, I'll look at it later. Okay. The electron transport chain. I do have written down stuff for this, but it's in the next slide because I'm just gonna go through the entire thing, explaining point by point, just because it's very... Actually, you know what I'm gonna do is... Hold on. I'm going to make sure this is recording, first of all. Okay. And then I'm just gonna change how I'm streaming so that I can actually point to stuff and not just go through this whole thing going crazy. Okay. Can you see my cursor now? Are we good? Are we good? Can we okay, good. Okay. So, the electron transport chain is probably one of the most annoying parts of cell respiration and stuff, but once you start to get it, it'll simplify down. So in the electron transport chain, now of course this is occurring in the mitochondria, so this phospholipid bilayer, recalled to unit 1, is the inner mitochondrial membrane. So this is the intermembrane space, this is the matrix, and that's the matrix. So the matrix is the inside of the mitochondria. I think I have it. Yeah, so this part, the dotted parts, are all the inside of the mitochondria. That's the matrix. These are cristae, and the two membranes here, this curve is what's shown in the electron transport here. So this intermembrane space is intentional. The cristae are, or like even this, it's intentionally that small because it helps in creating the concentration gradient. So of course within this bilayer, there's integral proteins. This is all the recall to unit 1. There's integral proteins, there's peripheral proteins, which all act as carriers of electrons in this situation. So first of all, in the biggest, not the biggest, one of the three biggest, there's 3 big membrane proteins, and they're that way because they also act as proton pumps. So in the first one, reduced NAD deposits, reduced NAD gets converted into NAD and H plus ions. The H plus ions are pumped through, and the electrons that are released from the reduced NAD are then transferred through the proton. The energy comes from the reduced NAD. So basically, reduced NAD gives the H plus ions, it gives the electrons, 2 electrons, and it gives the energy required to be able to pump the H plus from the matrix into the intermembrane space. And then the 2 electrons move their way into the other integral protein, and then after this step is where FAD joins in. Reduced FAD will deposit electrons as well. And then, oh, actually, sorry, I got this wrong. NAD doesn't supply any specific electrons. They just supply the energy. And then the electrons are supplied by the FAD, I believe. Feel free to correct me if I'm wrong. The 2 electrons and then move into the membrane protein here. the integral membrane protein here, and then it gets transferred to the next proton pump, at which point, again, the energy from these electrons will then pump more H plus ions into the intermembrane space. And then they, of course, travel. This is a peripheral protein, and then travel to the last membrane protein, at which point another H plus ion, more H plus ions are pumped through. And also, this is where the 2 electrons that are released from FAD, reduced FAD, get used up with oxygen, some of the hydrogen ions, to convert into water. So when you do the equation for aerobic respiration, we always say C6H12O6 plus 6O2 gives 6CO2 plus 6H2O. Now the H2O, this is where the H2O comes in. The electron gets accepted by the oxygen and the hydrogen protons to convert into H2O. After this happens, we look back to all the transfers that have happened before this. All the proton pumps have led H plus ions against the concentration gradient because this place is already concentrated enough, but it obviously uses up some of the energy to push against the concentration gradient, get the H plus ions into the intermembrane space, increase the concentration here, and then eventually get passed through ATP synthase to be able to convert ADP and another phosphate molecule into ATP. The adaptation of having cristae and having very thin intermembrane space is because of this specific phenomenon. This is called chemiosmosis, where because of the pumping of H plus ions within the intermembrane space and the increase of concentration gradient here, they have to move out through here, assist the movement of ATP synthase, which essentially creates enough energy to be able to convert ADP and a phosphate into ATP. Does everyone understand that? I mean, I will of course go through specific ones also, but are we good in terms of general understanding of it? God, so annoying. What's the circle 2 electrons again? Okay, wait, hold on. I'm re-sharing at this point. Okay, so the 2 electrons are basically just used to deposit into oxygen. The 2 electrons that are in, like the circles that are there, are all just membrane proteins. So they're either peripheral proteins, they're either integral proteins. If you recall back to unit 1.3 is membrane structure, we learn about that there. Yes, I can explain chemiosmosis again. Yes, that circle is just a peripheral protein. It's just what moves the proteins along the layer, move the electrons along the phospholipid bilayer. Because the electrons wouldn't be able to be carried just through the layer itself. The proteins are able to carry the electrons. You don't need to know why, you just need to know that it happens. Okay, the way chemiosmosis works is at every proton pump, there's enough energy due to the movement of the electrons. There's enough energy to push H plus ions that already exist in the matrix into the intermembrane space, which obviously there's an equilibrium between them, but H plus ions are pushed against the concentration gradient to be able to get pushed into the intermembrane space. So you can essentially think of it like when you push down on the top of a suitcase to be able to close it, that's essentially what's happening to all the H plus ions. It's getting pushed into the intermembrane space, even though it's probably very full already. Because they're so full, because the area is so full with H plus ions, because the intermembrane space is so narrow, all of that movement will then cause a concentration gradient again between the matrix and the intermembrane space, but they can't escape through the proton pumps because the proton pumps have to be activated by energy. So then they go through the next space available, which is through the gap in the ATP synthase, which allows the ATP synthase to move in circular motion, I believe, to be able to generate enough energy to convert ADP to ATP. Yeah, it actually spins. It's like it's a cylinder, and it actually physically spins because of the really fast movement of H plus ions. Because you can notice it's a very narrow like pathway, so it causes it to spin. It's actually really funny to think. Yes. Okay, that's the electron transport chain. So again, in a very simple way, this is of course the diagram, and which, by the way, you can draw the diagram in the exam. Of course, you don't have to draw the entire bilayer, you just draw it as just a layer, but you can, if you want, draw the diagram in the exam. It helps supplement your answers, basically. You don't have to, again, but it works well sometimes if you have enough time. Okay, so in the most simplest way possible, reduced NAD will turn into NAD and give electrons. I don't think it's electrons, actually. I think that's just energy. That's my bad. I'll change that actually right now, just so I don't spread false news. Okay. So reduced NAD will convert into NAD, or if you want to think about it in terms of H, NADH will convert into NAD plus, and that is what will provide enough energy for the first proton pump to push H plus ions. In another peripheral protein, FAD will deposit 2 electrons, which is what cause proton pumping at 2 stages. The 2 proton pumps after the fact, after the deposit, but NAD, the energy of NAD and the electrons of NAD cause proton pumping at all 3. Oxygen is only present in our entire process of cell respiration to be able to accept those 2 electrons. That's the only reason they're there. If there was no oxidation, the electrons would build up and the NAD would no longer be able to be reduced, which will just stop the entire process and the entire movement, and therefore you will stop breathing, which is why you can't not breathing, you stop respirating, which is why we can't live without oxygen. The oxygen play a really integral role even though they only appear once. It's very interesting. So of course, oxygen accepts the electron. It also binds together with H plus ions to convert into H2O, which is water. And then, of course, one of the most important parts, ADP will convert back into ATP, which, if anyone cares to know, the entire process of aerobic respiration, the entire process from start to finish, we say creates 38 ATP. While anaerobic only creates 2, obviously, because it converts when it goes through glycolysis, it only releases 2 ATP, which is theoretically what we say. So chemiosmosis, if you want a simple definition, is the generation of ATP using energy released by the movement of hydrogen ions across a membrane. Are we good with the ETC? Because I think the next one is an activity. I cannot speak anymore. Yeah. Any questions? Any at all? Okay, I see no one typing, so I'm just gonna assume there's no questions. If you have any questions, of course, you can always put it in chat and you can always ping me. Okay, so this is a mitochondria. What I want everyone to do is name each structure from A to F, label them basically, but also tell me what adaptation they have which helps cell respiration. So I want basically an answer. So I'll give you the answer for A. For example, that's the outer mitochondrial membrane, what it does, and how it helps aerobic respiration. This would be a sample answer. Of course, you don't have to write every single thing. I just want a general answer. If everyone could please do that for B to F. And by everyone, I mean the 3 of you who would probably do it. Which I don't mind at all, by the way. It's fine. Is anyone alive anymore? Is everyone just gone? Did Ari finally get their momos? Yes, alive people. If you want to answer the question, go for it. Just spoiler your answers so that we don't spoil it for people who want to try. I choose to believe that Ari finally got their momos. That is my choice. But yeah, I'll just wait until... I'll wait for, let's say, 7 minutes if everyone wants to attempt it. For a reason. Is anyone attempting the question? No? Are we just all tired? Which is fair. Again. But if no one wants to attempt it, I can just move on. I'm okay with it. Yeah, fair. It's completely fair. It's okay. I will explain it. I will go through it. I just wanted to do it as an activity because it's fun. For me, personally. Okay, so. The matrix. The second one is the matrix. So the matrix is what contains the enzymes for both the Krebs cycle and the link reaction, because that's where both of those processes happen. The small dots are the 70S ribosomes. That doesn't really matter. You don't need to know that one. That's my bad. D is space between inner and outer membrane. So this is where the chemiosmosis and intermembrane space comes in. Since the intermembrane space is so small, this the high proton concentration can be easily formed in chemiosmosis. So this adaptation is the most important one. The next is cristae. This is kind of part of intermembrane space, and it basically increases the surface area for oxidative phosphorylation, because oxidative phosphorylation happens really close to the membrane, and by increasing the surface area, it just increases how much happens at one time. The last one is inner mitochondrial membrane. By having two membranes, the inner membrane can have proton pumps that obviously create the electron transport chain, as a whole. Okay, we're at a break. The challenge is endoplasmic reticulum is not a bad shout, but this was a mitochondria. It's not a cell, so yeah. ER is not in the Golgi apparatus. They're two completely different things. ER is close to the nucleus. That's what usually has ribosomes, and that's what's used for proton synthesis. Golgi apparatus is for transport, so it basically creates vesicles that can leave the cell and basically causes exocytosis. That's all unit 1, I think. But yeah, anyway, so this time's challenge is beta D-glucose if anyone wants to try. If you don't, I don't stress too much about it. I'm just gonna give myself a M25 syllabus is weird, to be fair, so the fact that you're even here is good enough. Yeah, I'm gonna take a 5-minute break so I can drink water and be at peace. But yeah, if you want to take on the challenge, I call it a challenge, you need to know it for the exams. If you want to try it, go for it. If you don't, completely fine with me. Don't stress. I assume a lot of people are just going to sleep because it's so late, which is kind of on me for delaying it so much, but it's 12:38 a.m. Yeah, it's fine. I have a fucked up sleep schedule to begin with, so it's fine. The beta want to beat alpha, says the OH group. It's okay. There's a lot of people in the chat. If you scroll up, that's also the way a lot of people remember it. So it's not a bad way to remember it as long as it sticks in your brain genuinely. Up, down, up, down. Yeah, that's right, Lavender. Again, the only thing is on the completely right side, it's OH and the bottom group is an H. That O is the top right corner. Yeah, so that O will be an H, and then the top one that's empty is the O. No stress. Don't stress. At least you made this mistake now and not in the exam. That's the way I always look at it. Any mistake you make right before the exam, at least you've made it before the exam and you realize that you did it wrong. So you can do it better. There's something I was gonna say about something. That's fine. You just sent it from your notes. You can't do that. The challenge was to draw it right now. On the spot. You can't just send it from the notes. That defeats the purpose. I could send all my very prettily taken notes throughout the year, but it defeats the point of the exercise. It's just because people wanted me to do Unit 2, but I didn't have the guts to do Unit 2, so this is my way of integrating Unit 2 into this. Okay. I'll give it like 3 more minutes if anyone else wants to answer. I'm just gonna drink water, and then we can keep going and dive into the wondrous world of photosynthesis. Unit 2 is pain, which is mainly the reason why I didn't want to do it, because Unit 2 is the annoying unit. Like, I get it, but it's just I'm so scared to teach it because there's so much that could go wrong that I would just prefer not to teach it and just let everyone be at this point. Okay, I'm just gonna keep going because I don't think anyone's gonna try, and it's very late anyway. Is the structure a part of the same unit? No, it's part of unit 2. This is unit 8. I just put it there because glucose is part of this unit, and people wanted to do unit 2, but I just didn't have the ability. Okay. Photosynthesis. We've gone from cell respiration to photosynthesis. This is the last bit, and then we're done with unit 8. And then I have more sessions up for tomorrow. If you want to look at the event section, you can. And then, obviously, I'll go through whatever this... What is tomorrow? I don't even remember which ones I scheduled tomorrow. Tomorrow is the rest of... Oh, tomorrow is the hard ones. So tomorrow is all of unit 6 and 11. Whatever is left, whatever has not been done. So it's all the animal physiology unit. And tomorrow is also unit 9, plants. If people are struggling. I'm doing both of those. Tomorrow. I don't know if everyone does option C. I just do option C because I that's what our school chose. Our class chose. So that's the only one I can teach. I can't teach anything else because I haven't learned it myself. Yeah, fair. I feel like most people choose option C because it's the easier one of all of them. I'd say it's the easiest out of all of them because obviously option D is just all physiology, I think. And option A is all neuro, and option B is biochem. I don't remember, honestly. I don't know what option B is. Option C is ecology, yes. Biotech, there you go. So the thing is that they've created options really well in terms of spreading them out depending on what routes people take. Doesn't M25 have to do all the options but a little bit? Or at least that's what I heard from kids in my school. Yeah, they've made it crazy for you guys, but good luck, I suppose. Yeah, same. I'm glad I'm getting out. Jesus Christ. You are a guinea pig. I'm providing you with luck. Oh, Yob, I have a task for you. Draw beta D glucose. I'm not gonna start photosynthesis until you draw beta D glucose since you're back. Yes, I was waiting. Hey, you were able to do alpha pretty well, so you can do beta too. There's a joke somewhere lying under there. Please, no one make it. No, Lake, absolutely not. Part of the English test where you could bring 3 books? Huh? What do you mean? I was not part of that test. I don't think. I didn't read the instructions. The question I did had referring to 2 books, so then I just did. Anyway, we're not allowed to discuss the exams. I'm not gonna go there. I respect all the mods too much to do that today. Yeah, fair. Anyway, let's not discuss the test, guys, so that we don't cause problems with the mods. Interesting. That's very interesting. It's okay, y'all. You don't have to do it. I was joking. It's okay. You don't have to do it. But basically, there have to be 4 OHs, and one of the O's is in the ring. That's always there. Y'all, by the way. Ooh, okay. So, Oh, this is not bad at all. You are also missing you don't back down. So true. Okay, so to fix the issues you have here, the O is perfectly right. H, O, down. It's up, down, right? So it's the H and OH on the right will be switched. And then the OH is there, but there's going to be an H on top of that. And then the one on the left is fine. And then the one on the furthest left, there's an OH at the bottom. And on the right, they're switched. That's it. It's all right, though. And your CH2OH is just CH2... Is it CH2OH? It is CH2OH. See, 1, 2, 3, 4, 5, 6. It is CH2OH. Okay. Yeah. Got it. I got it on my own, over my own head there. Anyway. Okay. Let's go into photosynthesis. Photosynthesis. So the first thing you need to know is about chlorophyll and the photosystems. Who's asking me if I was going to go through photosystems? Whoever it was, we're here now. Chlorophyll and other pigments absorb light because they cause an electron in the pigment molecule to be raised to a higher energy level. Again, if people do chemistry, you know this. You just know this. But if you don't stress. It's just a thing to know. I don't think they ever asked this, but I put it there anyway, just in case they test us this year. Chlorophyll and other pigments can absorb certain wavelengths of light because an electron is moved to a higher energy level, and therefore it absorbs light. It's the absorption thing. The light energy is converted to chemical energy, which is then held by that excited electron. And the main photosynthetic pigment in plants is chlorophyll. And chlorophyll molecules in the chloroplast are a part of large groups of pigment molecules. And the entire group of pigment molecules is called a photosystem. That's what a photosystem is. So the photosystem is basically a group of pigments that work together to absorb light energy. And all of the pigments can absorb the light, but the photons of light have to reach the chlorophyll molecule at the reaction center of the photosystem to be able to pass on through the electron transport chain. Electrons cannot be transported by any other pigment other than chlorophyll, which is what makes chlorophyll so special. All of the pigments combined, the photosystem in general, is what will absorb the electrons or the energy, photon, sorry. But then what makes them able to travel is chlorophyll. There are two types of photosystems, photosystem 1 and photosystem 2, which, by the way, is reversed because photosystem 2 comes before photosystem 1, which makes no fucking sense, but we're here. There are two types of photosystems, photosystems 1 and 2, and they're located in different parts of the thylakoid membranes. Thylakoid membranes are part of the thylakoid stacks, which is part of the chloroplasts. Yes, you do need to know the wavelengths of chlorophyll A and chlorophyll B. I'm not talking about it today because my brain will not function enough for me to discuss this today. I think this is part of chapter 9, so I might talk about it tomorrow. I don't remember. Honestly, don't remember. Light-dependent reaction. This is very, 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 very similar to the electron transport chain in cellular respiration, but instead of proton pumps, in this you have photosystems. You do have proton pumps, but you also have photosystems. So it begins. I'm gonna change the way I am presenting this again, just so I can point, because I really cannot point if I'm just sharing one thing. It's so annoying. Anyway. Okay, so it starts over here. This is the photosystem 2. This is where it first starts. So the first step is photolysis, where Water is split into oxygen and hydrogen to release 2 electrons, which obviously photosystem 2 absorb. But the energy required to break the H2O bonds is what the photons are for. So photosystem 2 will absorb light energy, convert it to chemical energy, break the H2O bonds down, and then obviously use it as electrons. So electrons act as energy here. And then from the photosystem 2, it obviously reaches the control center of the photosystem 2, or the, what's it called, action center. Sorry, I literally talked about this just now and then I forgot. Reaction center. Into the reaction center, the electrons will reach the reaction center, and then because of the chlorophyll, will get passed on through the membrane proteins. The first one is just an integral protein, and then the next one is a proton pump, which again will pump protons from the stroma, which again we will talk about structures here, but we'll basically pump the stroma, pump protons from the stroma into the thylakoid space. That happens here, and then it'll pump 2 protons per pump. The 2 electrons will get passed on again, passed through photosystem 1, and then passed through electrons again, and then again, until it reaches a integral protein where NADP and the H plus ions in the stroma will be reduced together with electrons to convert into reduced NADP. Again, similar to cellular respiration and the chemiosmosis there, H plus ions are pumped through this proton pump. However, it's different in this situation because there's only one proton pump that pumps all the H plus protons. In cellular respiration, of course, there were 3. That's the difference here. And then, of course, because of the same phenomenon, an abundance of H plus ions will be formed over here, which will then pump through ATP synthase, convert ADP into ATP, which is then later used on in the Calvin cycle. Does everyone understand the light-dependent reaction? Is photosystem 1 and light-dependent reaction the same? Photosystem 1 and photosystem 2 are both parts of light-dependent reactions. Hold on, I need to freaking... Work. They get all excited in 1 and 2 from light, so they start running. Yes, that is true. Anything in biochem that has photo in it means light, and lysis means breaking. Yes, that is true. Okay, so in an easier way to explain this, let me just check one thing before I start explaining, just because I am also just wanting to make sure one thing. Okay. Yeah, so in photosystem 1, again, 2 excited electrons are passed through. This makes them more give them more energy because of the absorption of light, of course, and the photons. And then because they have more energy, they're more reactable to NADP and to H plus. So both photosystem 1 and photosystem 2 absorb light, but photosystem 2 is where the photolysis happens and where the first electrons are accepted. But then photosystem 1 is basically where the pair of excited electrons is emitted from, and then it passes through electron acceptors. So at the end of that chain is the NADP. Okay. So again, the ETC and chemiosmosis, this is ETC and chemiosmosis in the context of photosynthesis. So the first one is photolysis, where water converts into hydrogen and oxygen, releasing two electrons. These two electrons from photosystem 2 is passed on to a chain of carriers. The production of ATP is called photophosphorylation. We talked about oxidative phosphorylation. This is photophosphorylation, because oxidative phosphorylation is when it happens because of oxidation. This happens because of the presence of photons, and energy. The energy that is used is obtained by the absorption of light. That's why it's called photophosphorylation. At the end of the chain of carriers, the electrons are passed to photosystem 1, which then in the reaction center creates more excitement for the electrons and therefore leads it through a chain of acceptors, one of which that we learn about is NADP. So the three main conversions that occur throughout the photosynthesis ETC is the splitting of H2O into O2, 2 electrons and 2 hydrogen protons, NADP and 2 electrons, which then reduces the NADP, and then ADP and another phosphate turns into ATP. Basically, it's mostly the reverse of cell respiration, but there's still some differences. Of course, there's no FAD involved here, and there's fewer steps. Okay, I will be right back because I'm getting a call. BRB. Okay, sorry about that. I am back. Your class is not going over photosystems. You will eventually go over it. Don't stress. You will make it. I like how we've gone from... We were at 23 people. Now we're at 9. Which, fair enough. Who wants to listen to one person drone on for like 3 hours? But here we are. 3 hours? It's only been 2 actually this time. Which is surprising, because it took me a lot longer for the other ones. This is an interactive podcast. I'm telling you, this is basically a stream. I just don't show my face. And I'm talking about biology. I am not, but thank you. Okay. So, if people want to attempt this, again, no obligation to whatsoever, because God knows no one's brain works this late at night. If you want to go through what A, B, C, D, and D, E, and F are, you can just state what they are. I'm not gonna ask you to say what the adaptations are, because I'll explain it anyway, so there's no point in you writing it and then me just being like, okay, yeah, by the way, it's this. Yeah, but if you want to label it, please go for it. Does anyone want to do it? It's not a cell, it's the chloroplast. It's the chloroplast. You should be able to identify that, by the way. Yes, no shame in trying at all. And if you don't want to try, there's no shame in that either, please. I understand. Believe me, I understand. Oh, by the way, while I wait for people to respond, are people doing prom and stuff? This is because I feel like everyone here is M24 except you, Lake, but it's okay. I'll accept that. But yeah. When's prom? When's grad? I also am the one who was setting it up. Believe me, I was so stressed. I spent the last year just running back and forth in my school because no one would do anything. This is turning into a podcast now. Whoever's gonna get this, whoever's actually gonna look at the 2 hours worth of this recording is in for a ride. Because they're gonna go through so much bio, but at the same time, so much just me talking. It's so weird because I'm like, graduation is in like 2 weeks. Like what? No, it's okay. Don't stress at all. If you don't want to answer it, you don't have to. Seriously. My brain is barely working and I'm supposed to be teaching, so it's okay. Oh God. Good luck to you. But I'm glad you guys got it really quickly. There was a liquidity issue in ours too, but I was the one organizing all the fundraising, so I didn't let it happen. It was the most stressful year I've had. It was so bad. All the time while doing my IAs and EEs. It was fun. Okay. I'm not gonna read the answers. You can read the answers as I go through them. Sorry, I'm not going to correct the answers. You can correct the answers as I go through them. A is the granum. It is a stack of thylakoids for absorption of as many of the available photons of light as possible. Because they're in stacks, they increase surface area, therefore increasing how many photons are absorbed. B, thylakoid space. So each of them is a thylakoid space with a very small volume. So it can create a very steep proton gradient. So it works the same way as the intermembrane space in the mitochondria. This one is stroma. So the stroma is basically what the matrix is, or what the cytoplasm is in relation to the mitochondria in the cell. That usually has all the enzymes for the Calvin cycle, which we will go through next, and also has ribosomes and rubisco and naked DNA. So there's more synthesis of proteins and stuff within the chloroplast. Again, if you recall back to, I'm gonna say Unit 1 and the endosymbiotic theory, then you'll know why it can create its own proteins. The lipid droplet is just that. You genuinely don't need to know that. You just need to know that it can have lipids within it. So there's an envelope around the chloroplast which creates a compartment in which the enzymes can be concentrated. So obviously it's a double membraned organelle because of the endosymbiotic theory, and that means that it basically creates a fixed volume within which all the enzymes and all the components of photosynthesis are concentrated. E is a starch grain. Sorry, it's overlapped a little bit. E is a starch grain, which is created for storage of carbohydrates by photosynthesis until it's exported from the chloroplast. So when photosynthesis happens and when carbohydrates are created within the chloroplast, we will talk about this during the Calvin cycle also, but within the chloroplast, it can, a lot of the triose phosphate can link together to make glucose, and then all the glucose can link together within the chloroplast to make starch. Starch is how carbohydrates are primarily stored in plants, which is why you can see the starch grain within the chloroplast here. And the last one is the thylakoid membranes themselves. The membranes have a large surface area, and they also provide a site for the electron transport chain. Are we good? You are very good at bio. I support you 100%. Okay. The light independent reaction. So we went through the light dependent reactions already. This is the independent. This is basically the Calvin cycle. This is what happens. So, to go through the theory part of it, there's again three basic reactions that happens. There's carboxylation. So this is the opposite of decarboxylation. Decarboxylation was when carbon dioxide was removed. Carboxylation is the complete opposite of that, where carbon dioxide is fixed into the cycle to create glycerate 3-phosphate, also known as G3P. Another thing, just to remind you, never use GP. Always say G3P, because GP can mean a lot of other things. Because glucose phosphate is also a thing. So never say GP, it's G3P. So carboxylation is when ribulose biphosphate, or RUBP, which is created within the Calvin cycle itself, joins with carbon dioxide, which, by the way, is fixed through Rubisco, which is a protein enzyme. No, protein enzyme. What is Rubisco? Enzyme. It's an enzyme. Rubisco is an enzyme that you learn about in Unit 2 when you learn about proteins and enzymes and their specific functions. So Rubisco is used here to fix carbon dioxide into the Calvin cycle and create glycerate 3-phosphate. That glycerate 3-phosphate is then used in the main part, which is the synthesis of the carbohydrate itself. The G3P and some hydrogen atoms, I call them atoms, I think they're supposed to be protons, they join together to make TP, or triose phosphate. Now, triose phosphate we have encountered before in the link reaction, where hold on, let's go back just so we can see where we encountered it before, because now even I'm forgetting. Where are you, triose phosphate? No, we didn't. Sorry, my bad. Did we? Is it just me? Am I going dumb? I might be. It's okay. Anyway, even if we haven't looked at it before, triose phosphate is TP. That is what will link together to make glucose. So there, for every ribulose biphosphate, there's I'm gonna skip this for now and then go into this image just so it's easier for me to reference. For every ribulose biphosphate, which by the way is 5 carbons, it joins with 1 carbon dioxide, which makes it 6 carbons. Carbon fixation, the enzyme is Rubisco, makes it 6, which then divides into 2 glycerate 3-phosphate, which then go through the process of breaking down ATP into ADP and P, and receiving the hydrogens from NADP to reduce NADP to NADP, and through accepting the electrons from ATP and accepting the hydrogens from the NADP, it then creates into triose phosphate. This triose phosphate, so 1/6 of triose phosphate is what's used to produce glucose phosphate. So basically, two triose phosphates, when they are linked together, create glucose phosphate, but only 1/6 of all the triose phosphate that's created is used to produce the glucose phosphate. 5 out of 6 of those are what's used in used to regenerate RUBP by creating by breaking down ATP into ADP again. Is there a link to this presentation anywhere? I will put it in the chat after this session. I will also post a recording of this if it recorded at the end of this entire session, which, by the way, the recording and the presentation will get to you, but probably not in the next few hours. So just be warned, it won't be here probably until tomorrow. And by tomorrow, I mean tomorrow for me. I don't know what time it is for people, but it's just, I will try my best to upload the recording. It just won't be as quick, which I apologize for. I'm tired. It's not going to work well. Do we understand the Calvin cycle? Do we need me to go through anything? So, okay, before I look at if people have questions. So 1 out of 6 of triose phosphate is used to form several different types of carbohydrates. Glucose phosphate is created by linking 2 triose phosphates together, of course, because triose phosphate is 3 carbons. Linking 2 of those will give you 6 carbons. But more importantly, starch is created by linking multiple glucose phosphates together. So this happens in the not matrix, in the stroma of the chloroplast after the entire process of Calvin cycle has occurred. Calvin cycle also occurs in the stroma, and then as soon as it occurs, then the glucose can start linking together to form starch by condensation. And that's all. If you guys have any questions, you can ask me. If not, feel free to leave. I am letting all of you go. Oh, yes. Okay, sure. Thanks for reminding me, because I would have forgotten. Okay. So, in the light-dependent reactions, photolysis of water occurs in the thylakoid space. But how come the pumping of protons is from the stroma to the thylakoid space when in reality the thylakoid place is already rich? Okay, yes. So this is what I was talking about in terms of, I'm going to go back just so it's easier to reference. This is what I was talking about in terms of the proton pump requiring energy. Because this is happening in the thylakoid space, this place is already rich with hydrogen ions. But this pumps against the concentration gradient, which is something I mentioned for cell respiration but didn't for this, which is my bad. But basically, it pumps against the concentration gradient, even though this is at higher concentration than the outside. It pumps against just so this can become rich, even richer, and then therefore leave here by chemiosmosis. The entire function of this being rich in hydrogen and this pumping anyway is because it needs to have enough of a difference between the stroma and the thylakoid space to be able to diffuse through this through the ATP synthase. Does that make sense? I think the person who was asking the question isn't even here anymore, but Steph, if that makes sense, let me know. If it doesn't, I can repeat it again. Okay, I can repeat it more slowly. So generally speaking, we know that photolysis happens within the thylakoid space. So there's already H plus ions there. And there's H plus ions in the stroma as well. After photolysis happens, there is an increased concentration within the thylakoid space, but it's not enough to move the ATP synthase. The reason we need it to be more is because we needed to have enough of a concentration gradient that there's enough force happening when the H plus ions are moving. So when you move against the concentration gradient, it has to be through active transport. That's why it's through proton pumps. Now, you can never travel backwards from proton pumps. It's directed by the pump itself. So because of the energy that electrons leave as they transport through the electron transport chain, because of the energy, the proton has enough energy to be able to push H plus ions from the stroma into the thylakoid space. The reason they push further into the thylakoid space is to be able to create enough concentration and enough activity within the thylakoid space to be able to push through the ATP synthase and move it so that ATP can be created from ADP. That's the entire process, if that makes sense. And another reason why photolysis happens within the thylakoid space is because in the light dependent reaction and in the electron transport chain for photosynthesis, there's only one proton pump. While if you look back to the cell respiration electron transport chain, there's 3 different proton pumps. So it creates enough of a concentration gradient just by moving those 3 in, right? But in this, there's only one. So to be able to create enough of a resistance, they have to, photolysis also has to happen within the thylakoid space. I hope that makes sense. Okay, yeah. If anyone, I'm gonna just go back. If anyone has any questions, feel free to ask them. If you don't have any questions now, you're free to go. If you have any questions that come up later, you can always ping me in M24 biology, and I will try my best to answer the question. If I can't answer the question, of course, you can also ask Lipid, who's the other biology helper. Yeah, that's pretty much it for me. If you have any questions, seriously, do not hesitate to ask. You can also DM me at any time. My DMs are usually, I'm pretty sure, always open. DM me at any time, and I will do my very, very best to answer. Okay.